Hello children, welcome you all to the Form 7 biology session. Today I am going to discuss one of your first term content, microbiology, which comes under the SOW reference 6.1 to 6.5. First of all, let's see who are these bacteria and viruses. So bacteria and viruses are the main disease causing pathogens in humans. But as we all know, bacteria are the most abundant organism in this universe. So compared to the bacteria, viruses are very small organisms. But even there is a confusion whether these viruses are a living organism or a non-living thing. We can't distinguish viruses as a living thing or a non-living thing because the only metabolic process that viruses possess is the reproduction. That is only happening when viruses are present in host cells. But bacteria, they can reproduce in many places. And also these bacteria, basically they are called as prokaryotic organisms. They don't have an organized nucleus. But bacteria, they contain ribosomes. So they can perform protein synthesis. But viruses, they do not have these ribosomes and other cellular organelles like bacteria. So bacteria are living single-celled microorganisms, while viruses are technically non-living. So the structure of viruses is much more simpler and smaller compared to the bacteria. So bacteria can reproduce in many places and they can perform binary fission where viruses can only replicate their proteins by invading a host cell. Then these viruses, they are taking the control of the protein synthesis mechanism of the host cell. Now we will see the viral structure. So basically, viruses, they contain either DNA or RNA. So they have a capsid, which is a protein coat surrounding the genetic material, and also they have some attachment proteins. So these stick out of, from the capsid and are used for the attachment to the host cell. And also they contain enzymes. So many viruses, they contain enzyme called reverse transcriptase. So that is used to convert their RNA into DNA. So which can be inserted into the host cell's genome. So viral proteins are transcribed. So viruses, they have different shapes. So you can see here helical shape, icosahedral, and some are having complex structures. Now we are moving to the life cycle of virus. So it can be explained via two ways. There are two cycles actually, lytic cycle and lysogenic cycle. Lytic cycle we can call lytic cycle. So now we will see what are happening in the lytic cycle. Initially, the virus or the phage infects a host cell. So then after, the DNA of the phage circularizes, remaining separates from the host DNA. So you can see here the green color one, that is the DNA of the virus. Then after, the phage DNA replicates and phage proteins are made. Now, new phage particles are assembled in the host cell. So now you can see the replicates. Then after, what happens is, the cell membrane of the host cell, it ruptures. The cell lyses, so releasing the phages. So these phages, they can infect other host cells as well. Now we are moving to the lysogenic cycle. So here also initially the phage infects the cell. Then after, the phage DNA becomes incorporated into the host genome. After that, the cell divides and the phage DNA is passed into the daughter cells. So here, not like in lytic cycle, the phage DNA is incorporated to the host genome. So under stressful conditions, the phage DNA is excised from the bacterial chromosomes and enter to the lytic cycle that we have explained earlier. So now this is the life cycle of the HIV virus. HIV. So initially the receptor binding, you can see where these attachment proteins. Then after the membrane fusion and entry. Third one, uncoating and reverse transcription. Further the nuclear uptake. Then after the integration to the host genome, transcription, RNA processing, and then the nuclear export. Finally, the translation, that is simply the protein synthesis mechanism. Then after the assembly, further the budding and releasing. This is the life cycle of HIV. Now we are moving to a separate section, culturing microorganisms. So culturing microorganisms, this is very important in microbiology. So we can obtain pure cultures, sometimes we can obtain mixed cultures. So pure cultures, they contain only one type of microorganism. But mixed cultures, we can obtain different types, kind of like a mixture of species. So when we are culturing microorganisms, we need to follow aseptic conditions, aseptic techniques in order to prevent the contamination. So basically we have to use sterile equipments. There are so many ways to sterilize these equipments. For an example, we can direct heat. We can apply direct heating, or else we can even use autoclaving, heating to high temperatures, especially for glasswares and other equipments. And even for media, we can perform autoclaving. And also before we are using these surfaces, we have to clean. So we can clean these surfaces like bench tops using ethanol. So the Bunsen burner we can use for direct flaming or direct heating, especially for the inoculation loops. Now you can see here in the diagrams, so a petri dish set up for culturing microorganisms. And in the second diagram, you can see the microorganisms which have cultured in a petri dish. But this is a mixed culture. We can obtain different types of colonies. And also, there are two different types of culturing, batch and continuous. Now we will see what is the continuous culturing and what is the batch culturing. So in batch culturing, the fermentation is carried out in a closed container, closed fermenter. Microorganisms and the nutrients are added and then left to grow for a certain time period. So further, we are not adding any nutrients and the products are removed at the end of the period. But in continuous culturing, so here we are using an open fermenter where the nutrients are replenished. And these are some of the important points that you need to remember. In order to maximize the yield, in order to increase the product, the temperature and the pH, these conditions should be properly maintained. And we have to make sure if we are using aerobic organisms, the oxygen supply should be properly controlled. Now we can see here the measuring the growth of cultures, measuring the growth rate. Here we can use cell counting method, serial dilution or dilution plating method, and we can just measure the mass and the turbidity measurements. So turbidity measurements we can perform using the equipment called as the spectrophotometer. Now initially we are moving to the cell counting method. You can see here this equipment we call hemocytometer. So using the hemocytometer, we can count the number of organisms. So in here you can see microscopic count. All cells in several large squares are counted and the numbers are averaged. The large square shown here has 4 bacterial cells. You can count here in this large square. And then the serial dilution. So here what we do is we are performing the dilution. We are diluting the plates. So here we can reduce the number because initially the large bottle, it contains a large amount of microorganisms. By performing this serial dilution, we can reduce the number. Then it's easy to culture and easy to count. Then we are moving to the turbidity measurements. So here we are using a spectrophotometer as shown in this diagram. So from here we are taking absorbance measurements. So this absorbance is directly proportional to the biomass, to the mass of the microorganism. So here we have to follow a law called Beer-Lambert law. According to the Beer-Lambert law, the absorbance measurements are directly proportional to the concentration of the substance present in a certain solution. But here as we use microorganisms in the solution, so that is directly proportional to the turbidity measurements, means the number of organisms present in the solution. So in the diagram now you can see there are 4 test tubes. So from here to here, from left to right, the turbidity increases. Increasing turbidity means the number of organisms present in the solution is higher. Now these are the phases of bacterial growth curve. So the y-axis is the number of bacteria in log scale, and x-axis, that is the time. So basically the bacterial growth curve, it is consisted of 4 phases. Lag phase, log phase. Log phase we call the exponential phase where the growth is higher, and the stationary phase, and finally the death phase. So this is for your extra knowledge. The plasma viral load and the CD4 lymphocyte predict simply how the lymphocytes are changing, the amount of lymphocytes are changing over years, over weeks, years, when a viral infection is there. So you can see here, blue color line and the red color line. 2 plots are there in the same graph. So here the primary infection. Then after, what happened to this? After the clinical latency, the opportunistic disease. And finally, when the number of copies of the RNA or the virus copies, virus DNA or RNA is increased, so what happens is, so the symptoms of AIDS and the death. So here we know HIV, that is a retrovirus. Retrovirus means RNA virus. It contains RNA as the genetic material. Now there are some questions. You can try these questions, and here even I have attached 2 links for 2 question papers related to the microbiology. You can try these at home, but we will try the second question. For each feature, place 1 cross in the appropriate box in each row to show whether it is found in all viruses, some viruses, or is not found in viruses. So now we will see features: cytoplasm, DNA, protein code. Cytoplasm, it's present in all viruses, some viruses, not found in viruses. Then DNA, whether it's found in all viruses, some viruses, or not found in viruses. Next one, protein code, present in all viruses, some viruses, or not found in viruses. First we will go for DNA. DNA or RNA. So DNA can be present in some viruses. Protein code also capsid, that is present in most of the viruses, all viruses. Cytoplasm. So what is your answer? We discussed that we can't distinguish virus as a living or a non-living thing. So cytoplasm is not found in viruses. Okay, right. But DNA, some viruses, protein code, all viruses. Right. Now you can do the other question, and also please download these 2 question papers and please do those at home. That is the end of the presentation. Thank you for listening.